Hello everyone, welcome to MISIANI'S lectures, and today we are going to biology, basic biology concepts, and today we are going to do the glycolytic pathway. And as we know, when we eat food rich in carbohydrates, and the main carbohydrates that we normally get in food is starch, and starch is normally broken down in the mouth by the aid of salivary amylase and amylase in our intestine to produce glucose. So the ultimate product that we normally get from carbohydrates is glucose. And today now we are going to see on how we break down glucose to produce energy, and that is the glycolytic pathway. So let's get into it. So we will be starting with glucose. From glucose, and we go steps down up to the final product that we know in the glycolysis. That is the glycolytic pathway. So we will start with glucose. Glucose will be the first precursor that we will get in the food after starch has been broken down. So glucose to glucose 6-phosphate. And as we know in biological systems, every reaction, there is always an enzyme that catalyzes the reaction. All biochemical processes contain substrates, enzymes, and the reactions that takes place, we always have enzymes and substrates. So in this glucose to glucose 6-phosphate, we will be having ATP getting into and ADP, ATP and ADP. So that is adenosine triphosphate. One phosphate will go to glucose and we get adenosine diphosphate. So the enzyme that catalyzes the reaction is hexokinase. Hexokinase, as we know, category of enzymes, we have kinases, isomerases, and as we know, kinases normally deal with transfer of phosphate groups from ATP. So we are going to the next. We are going next to the next reaction. From glucose 6-phosphate, we get fructose 6-phosphate. Fructose 6-phosphate. Fructose 6-phosphate is catalyzed by an enzyme that will change glucose, a 6-carbon molecule, to fructose, a 5 carbon molecule. So that is isomerase. Sorry about that. Isomerase. So from fructose 6-phosphate, we will go to fructose 1,6-biphosphate. So we had fructose 6-phosphate and we are going to fructose 1,6-biphosphate. That means we are adding another phosphate on the fructose molecule. So we will be spending another ATP molecule to get ADP. And as we know, enzymes that catalyze reaction involving phosphorylation are hexokinase. So that is fructose 6-phosphate kinase. So that one is a kinase enzyme. Phosphohexose kinase. So from here we have our fructose 6-phosphate. Let's continue with our pathway. So we will go to change this process, we will have to change from fructose 1,6-biphosphate. We have to change this into a 2,3 carbon molecule. That will be dihydroxyacetone 3-phosphate plus glycerolhaldehyde 3-phosphate. Dihydroxyacetone 3-phosphate and glycerolhaldehyde 3-phosphate. These are 3 carbon molecules, both of them. So we are changing from a 6 to a 3 carbon, and the enzyme that catalyzes the reaction is called aldolase. So from here, the fate of dihydroxyacetone 3-phosphate, this molecule is normally used or normally goes to biosynthesis of fatty acids, and it's normally used in synthesis of TAGs, triacyl glycerol, because we will get the glycerol molecule from here and it will combine with acetyl CoA and it will go to synthesis of TAGs, triacyl glycerol. So we will continue with the 2 molecules of glycerolhaldehyde 3-phosphate. So, and for this process, we will have to isomerize this molecule to get this is glycerolhaldehyde 3-phosphate. So we will go to, and we must know that these are 2 molecules of glycerolhaldehyde 3-phosphate. So from glycerolhaldehyde 3-phosphate, we will spend 2 phosphate molecules and NAD plus will get in, we will get NADPH. So as we know, NAD, this is nicotine adenine dinucleotide. These are oxidative coenzymes. So, and where NAD, we know the enzyme is glycerolhaldehyde (3-phosphate) dehydrogenase. Here, the product will be 3,1,3-phosphoglycerate. So we have 1,3-phosphoglycerate. We got 2 phosphates here with NAD catalyzing the reaction. NAD, we get an NADH. This one we know they are oxidative. During the oxidative phosphorylation, we get NADPH. So as the electron carrier. So we go forward and we get our first yield of ATP. So the next step, we will have 3-phosphoglycerate and ADP and ATP. So we get here a net gain of 2 ATP molecules, and the enzyme is phosphoglycerate kinase. kinase. Phosphoglycerate kinase, because of the phosphorylation of ADP to ATP. So we continue with our pathway. From 3-phosphoglycerate, we have to change the isomerism to 2-phosphoglycerate, and we will have our enzyme amutase because it changed the position. If you remember chemistry, there was a topic known as isomerism, changing from one position to another. That is amutase. And then we will go from 2-phosphoglycerate, we have phosphoenol pyruvate. Phosphoenol pyruvate, and then that is the enzyme is enolase. From phosphoenol pyruvate we get pyruvate, and we get our second yield of ATP. We get our second yield of ATP, ADP, ATP. That is 2 molecules of ATP, and we get pyruvate, being the last step in glycolysis. So, as we see in glycolysis, we used 2 molecules of ATP and we gained 4 molecules of ATP. So our net energy gain will be 2 ATP molecules. So the fate of pyruvate depends in whether it is aerobic or anaerobic respiration. If there is presence of oxygen, pyruvate will be decarboxylated to form acetyl-CoA, and acetyl-CoA will go to Krebs cycle or tricarboxylic acid cycle, whereby it will go to get to be further oxidized and yield more ATP molecules, and I will be covering that in the next video, whereby we will be talking about further breaking down of pyruvate. And if there is absence of oxygen, pyruvate will produce lactic acid by lactate dehydrogenase to produce lactic acid and CO2, and that is where we normally get muscle cramps and all that, because of anaerobic respiration. And I guess that will be the fate of pyruvate. It's either presence of oxygen or absence of oxygen. Thank you so much, and welcome, subscribe, and wait for the next video.